The passage introduces a new system about slab gel electrophoresis. This system can not only greatly reduce the chemical consumption, save experiment time, but also can realize real-time tracking DNA separation in the agarose gel by smartphone. Those three pictures represent a typical result after gel electrophoresis of 50, 100, and 1000 base pairs DNA ladder. Each DNA fragment was well separated.
Slab gel electrophoresis is the most effective way for DNA fragment separation, and thus is deemed versatile in biochemical and biological analysis. This time, I'll show you a new method about slab gel electrophoresis experiment. Okay, let's begin.
First, we weigh out 0.1 gram of agarose gel and put it into a flask. Add TBE buffer into the flask. The volume of the prepared solution should be not more than 1/3 of the capacity of the flask. Seal the flask with preservative film, and then the agarose buffer mixture was heated by a microwave. Prior to putting the flask into the microwave, we make some holes in the preservative film in case of explosion. Pour the melted agarose solution into the channels in the slab gel electrophoresis chip. Place the comb into the solution. The melted solution will cool down and become gel state at room temperature 3 minutes later. Take out the comb to create the wells and add TBE buffer to cover.